Good afternoon, everybody. Thank you for joining us this lunchtime for our Protect webinar today, where we'll be thinking about some of the risks that might be associated with food manufacturing. We'll just give it a couple of seconds to allow people to join. I know it's a common issue where people are meeting hopping. So what we thought we'd do by way of introduction is just for me to give a bit of an introduction to the Protect study before we move into the three substantive presentations. If you can make sure that you are on mute when the speakers are talking, that'd be hugely appreciated because it can be very distracting. And to flag up that there is a question and answer function at the bottom of your screen. You can ask questions at any point. We'll have time for some questions after each of the presentations, and then obviously a more substantive panel discussion at the end. So please do feel free to enter your questions at any point during the event. You can also like the questions, which moves them up the list. So please do use that function as well. Okay, so as I say, what I'd like to do to start with is just give you a very brief introduction to the Protect National Core Study and what we've been doing over the last 18 months or so to try and understand exactly what is going on with respect to transmission of this virus. And to do that, I just need to press that button. So the Protect study is part of a program of studies that were set up at the start of the pandemic to enable us to have some really rapid advice on key areas of concern when it came to SARS-CoV-2. Patrick Vallance established the program. There are 6 projects in all, and the intent, as I say, was to provide a rapid and dynamic response to the pandemic. The program is based on the sort of fundamental principle that there are 3 main routes of transmission, namely surface, airborne, and person to person. But more importantly, I think the program has identified that there are 3 key areas that we need to focus on to enable us to really get to grips with the transmission issue. And that boils down to understanding environments, thinking about how humans behave in those environments, and how the virus itself also behaves, both in the humans and the environments as well. And it's only by bringing those 3 different areas together that we really get a good handle on transmission events. The program started back in October 2020 and will finish in March 2023. And it's very much based on an understanding of the real world. One of the things that we were very keen to do is to provide insights perhaps that weren't available through traditional modeling routes or very focused laboratory type studies, but to go out there and collect information from real people in real workplaces. And hopefully you'll hear more about that during the presentations today. We also obviously wanted to make sure we were leaving a legacy for future pandemics. So that's also part of our process going forward. We have a number of themes underpinning the work, and you can see what they are in front of you. We'll hear from some of those themes today, particularly around the work we've been doing on outbreaks, and as I say, going into real workplaces and collecting real data from real people, but making sure that we are taking that information and using it in qualitative and quantitative models so that we are validating those models appropriately. We're getting more information from sectors. So obviously today we're talking about the food processing sector, but we've also looked at things like transport and how that might serve as a route for transmission. We've done work on experimental infections, so using animal models and human volunteer studies, where we've collected virus in both those situations to try and understand the temporal nature of virus emission and the variability in individuals of virus emission. Again, really important to help us understand how and when and where transmission occurs. We've also had to make sure that we've got the right tools to do that. So we've been working really hard to make sure that the measurement techniques are appropriate, that we can go and detect viral RNA in difficult places, and you'll hear some more about that today, and use that information again to inform our thinking about what actually has to happen for a transmission event to occur. And of course, critical to all of that is making sure that we synthesize that knowledge into actionable policy advice, and importantly as well, practical tools for people to use in real workplaces. We will be delivering this program over 3 years. We're currently coming to the end of our second year, and you can see that initially we've had to establish a core team because we were starting with a blank piece of paper. There really wasn't anything known about transmission in those early days, and we had to bring together a group of scientists and engineers from disparate backgrounds and different organizations across the academic sector and the government sector to enable us to really get to grips with this difficult problem. That enabled us then to really start to do the experimental work in the second year and think about the mix of activities that would give us the best information to understand transmission from a variety of different perspectives, focusing on that environmental, behavioral, and viral characteristic perspective, but really focused on that real world situation. As we move into our final year, we need obviously to conclude the research activity, but then to do a significant amount of stakeholder engagement so that we can work out exactly what it is that people want from this program. And if you haven't already been asked, please do contact us, because we're keen to hear some of the really practical questions that you still want answering. And one of the things that we are doing is currently synthesizing the output from a big survey that we've done of stakeholders to see if we can start to address those key questions in a format that provides people with the right information in the right way so that they can take appropriate action going forward. And then, of course, finally, we want to make sure that we've got a legacy program, and that will include making sure that we've identified the need to maintain this capability going forward, and to see how it might also help if you take COVID out of the equation and think about the benefits and routes to impact for other issues. For example, how you might maintain an infection resilient environment moving forward. You can see the amount of money and the number of partners, the number of projects and the number of researchers. And it's always difficult to pin down the number of publications because they are coming out thick and fast now. As I said, one of the challenges was starting with a blank piece of paper. You have to do the research to enable you to start to publish. And of course, we are now at the stage where those publications are really rattling through the system. We're just over halfway through the study, and it's important always to check that your key stakeholders are finding value in what you're doing. And you can see that both Patrick and Patrick's office are making good use of the information that we're providing. And certainly moving forward, how we feed into that wider legacy program and what we can learn more broadly from the work we've done when you take COVID out of the equation. And of course, all of this is only achieved through the efforts of a large group of individuals. And I want to specifically acknowledge the work of our theme leads, and Yiqun Chen, who's talking today, is one of those. And they've done a fantastic job at brigading together the researchers and the research organizations that you can also see on this slide to deliver what is without doubt one of the most extensive and important programs of work to understand transmission in a wide range of different environments. So that brings us on to today, and what we wanted to do today was to focus on food manufacturing. We have done work in that space. As I mentioned, one of our deep dives was to food manufacturing, and some of our outbreak investigations have been into food manufacturing. So it's a great pleasure to welcome three fantastic speakers today. Firstly, we're going to hear from Dr. Yiqun Chen, who is an HSE member of staff, a senior scientist who leads one of our hubs, particularly on the right evidence for the future, and she's been leading the theme on outbreaks. We'll then hear from Rob Johnston and Dr. Barry Atkinson from UK HSA, the UK Health Security Agency, who'll talk about some of the work they've been doing on surface sampling in food manufacturing processes for the virus. And then finally, we'll hear from Dr. Miranda Lowe, who's Head of Environmental and Public Health at the Institute of Occupational Medicine. And she's going to talk about some of the work that IOM have been doing, looking at the risks and experiences of the virus in the food and drink manufacturing sector. So hopefully what we'll be able to do at the end of that is have a good discussion about some of the things that we found. And of course, as I say, please do use the Q&A button to post questions into the system, and we'll take some questions after each of the presentations. You can do that at any point, and as I say, you can also use the like function to move questions up. So without further ado, I am going to introduce Dr. Yiqun Chen, who's now going to talk to us about the work that she's been doing, understanding what's been going on in the food and manufacturing sector from the outbreak investigation perspective. So Yiqun, thank you very much for joining us today, and over to you.
Thank you, Andrew. Yeah, I'm going to look at outbreaks in the food manufacturing sector, and my presentation is focusing on answering two questions. First, whether the food sector has increased risk of outbreak. If that's the case, secondly, what will be the contributing factors? We bring the data together to look at the outbreaks across different sectors, but we also look at evidence from international scientific literatures and supporting the evidence we collect from on-the-ground investigation of outbreaks in a meat processing plant. In a very early stage of the pandemic, we were able to analyzing the Public Health England's outbreak records, look at those outbreaks in different sectors and compare to the number of companies in those sectors and calculate the outbreak rates. You can see that food sectors have the highest outbreak rates. About just under 2 companies per 100 companies have outbreaks over the 5 months period. And when we look at across different English regions, food sectors consistently have high outbreak rates in most of the English regions, apart from East Midlands and North West, where warehouse and distribution centre has the highest outbreak rates than the food sector. That is a number of outbreaks, but within each outbreak, we also look at the number of positive cases. So within the food sectors, actually, the number of positive cases is about 2%, which is much lower than other settings like the close context or other non-food manufacturing sectors. So we were able to look at longer time periods about outbreaks that investigation that HSE has involved. At the national level, the outbreak investigations is led by the local public health, but when they consider that workplace transmissions could be a contributing factors, they will involve HSE inspectors. So over about 20 months, HSE has involved over 270 outbreaks. 20% of those outbreaks were in food manufacturing sectors. Using this data, we also found that food sector have highest outbreak rate. But when we look at over time, most of the outbreaks that HSE has involved in the investigation was before March 2021. So you will ask, maybe that is the bias because we focus on the outbreak response in those particularly high profile sectors. So we look at the news reports of outbreaks in workplace. We consistently collect information about outbreaks in a news report in workplaces. That 6% of those reports were related to food sectors, and using that data, food sector also have highest outbreak rates, even though there's only 22 outbreak reports in the news compared to, for example, 910 in the non-essentials sectors. And once again, most of the outbreak report in the news also before March 2021. So we look at this graph. This is outbreak rates using the HSE outbreak records and the news report, and compare the populations COVID cases. You can see that most of the outbreak reported or investigated were before or related to the first wave, before before March 2021, and also before the mass vaccination. In the population level, by June and July, about 50% of the general population were double jabbed. So we also look at the number of spot checked. So over the 20 months period, HSE has done or has spot check attempt in about 500,000 of the workplace, which covered a quarter of the workplace in Great Britain, with 50%, about 50% response rates. And with different stage of inspections, at the last stage is on the site inspections. And among those, about over 2% of the companies in food sectors have failed their inspections about the COVID-19 control. So another piece of information I thought would be useful is that concern data. This concern were submitted by public, by workers, by employers, and that is not influenced by HSE's operation or public health operation. So of those concerns, about 100,000 is from the manufacturing sector, who were not able to separate food and non-food, but it's just the manufacturing sector. They also have high proportions of the concern, that is red and amber. So based on the information I explained, that food manufacturing sectors do have a higher rate of outbreaks. So what is the risk factors could contribute to those outbreaks? We have done a systematic review of international scientific literature. We found 6 publications related to food sectors. They all in a meat processing plant. They describing the outbreaks occurred before February 2021. That's also related to the first wave. And early stage, they have identified several risk factors in these sectors, like high employee densities, and the specific work practice and the design of production line. Later on, they also look at environmental conditions like low temperature and the lack of ventilations could play a part in promote the virus transmission. When we look at the interventions, it's quite interesting to see that at the early stage of the pandemic, the testing were only for the symptomatic workers, and also face coverings was optional. But later on, with more scientific evidence, they started to look at the control measure related to ventilation, and later on in the vaccination in controlling outbreak. We also look at the records. I explained that HSE involved in public health outbreak investigation in the workplaces. We have over 700 outbreak records. We review those records. We have about 20 samples of those records from food sectors, and that is not unique to meat processing. And we highlight the theme of the risk factors, mostly is related to social distancing, cleaning, and shared transport. And in terms of control practice, mostly focusing on testing, contact tracing, and the staff communication. So I want to spend a bit more time looking at our on-the-ground outbreak investigation. So this is the COVID out study, part of theme 1, protect. We go onto the outbreak settings as soon as we have the notification of the outbreaks, to collect data to understand the environmental conditions and also their control measures to help us to understand the risk factors of the outbreak, as well as to understand the relative importance of different transmission routes. So for this study, we have recruited 3 food production manufacturing sector companies, one related to Delta variant outbreaks, and this is what I'm going to describe later, and two relevant to Omicron variants that we still are collecting data from those two sites. So this is the outbreaks in a large meat processing plant that we have investigated. You can see that this company have outbreaks early on in January, and they have a second outbreaks starting from the end of May, where the Delta variant started to dominate the population. We know that because most of the early cases from this site had S gene positive, that is a surrogate to understand is related to the Delta variant. And at the beginning of the outbreaks, the local population's infection rate is about 20 per 100,000. But at the end of outbreak, the end of June, the local transmission rate is 200 per 100,000. You can see that the local transmission will influence workplace transmission. And this workplace have in place the lateral flow test, daily lateral flow test, no, twice weekly lateral flow test, and have their test centers from March, but the outbreaks still happen. And after the outbreaks occur, they have a 1 week PCR mobile unit in place, and also the vaccination starting and at the end of June. So it just shows that they did control the outbreak quite successfully. And this large workplace, I just want to give a basic information about the workforce. It's about 1,500 workers, have two shifts, day shift and night shift. And in the middle, they have a hygiene shift that focusing on the cleaning of the production area. And people also work rotating day shift and night shift as well. This is the timeline of the outbreaks. From day one, the first case were identified until day 30. You can see that most of the cases were among the night shift workers, with a small number in the day shift workers. You can also see that there are partners who work in night shift and day shift have positive COVID-19, and you have partners both working night shift. And you also have people that have relatives living with people who test positive, but mostly 2 days after the workers test positive. So it looks like there is a spreading risk within the night shift workers. The attack rate in the night shift is 9% compared to 1.8% in day shift. I have to say this is very underestimated, because the denominator, the number of workers, I put everyone in the book as a denominator. And we know that actually it's much less number of people working in a night shift, and probably a large proportion or significant proportion of them working part-time. And among them, including people who are coach drivers who do the staff transportation, but also have people work in the office. But most of the cases come from the production line. All areas of production lines have people test positive. So it's quite a widespread within the workplace. And this is the map of the workplace. And you can see that this is the car park, and people come in with two entrances, and they have men locker area and boot wash. And there's two link production area. Production one, area one is meat cutting. And you can see that on the right-hand side, this is the chicken factory. The chicken, the intake areas, they put the chicken into the production line. And this is the cutting and production preparation areas where they cut the chicken into parts and before they put in the conveyor belt. And do further processing. At the end of the conveyor belt, it will become a packing area. And on the left-hand side is areas 2, is minced meat, how they separate the meat from the bone. And as much colder areas on the left-hand side. They have their own dispatch area. And the employer also put in extra smoking shelters, locker rooms to space staff out. The production areas is 700 square meter, and the maximum number of workers work per shift is 450. So you can see it's a very high density of employees. And this blue circle is air handling unit, and those two are only for the non-production area. The production area 1 air handling unit, we cannot identify it, even we did investigate with the employer. And the air handling unit for area 2 were not operational. We were informed by the ventilation contractor. In this company, the ventilation system were managed by the contractor that is not belong to the company. That's the issue that we need to discuss later. Looking at environmental condition, this is the table to look at the CO2 measurements. This is the continual measurement results, temperature, humidity, and the noise level. You can see the CO2 level are very high in the production area, but we know that they use CO2 for packing process. So we find it very difficult to draw conclusion. I list these areas in a distance between, so the top areas is related to the production area. The bottom actually is the bottom canteen and the locker room is away from the production area, should not be affected by the CO2 using the packaging process, but we still find that the main locker room have higher CO2 level, and that is related to the shift starting and finish time. And the temperature is very low in the production areas, 4.5 degrees, and very high humidity, and the noise level in the production areas are very high. And the company measurements of the noise level in the production areas were even higher. That caused problem in the communication and the social distancing. So I just want to give you an example of these continual measurements. This is the top one is the temperature and humidity. We said that the production line have a very low temperature, but could be even lower than 4 degrees, because the higher temperature which were related to the cleaning process. So you can see that production workers are working in a very high humidity, a very low temperature. And the lower graph shows you the CO2 measurements, showing that high CO2 level in the locker rooms, relevance to the shift pattern. And I don't expect you to read all this, but I just want to highlight those risk factors we observe in this workplace. First is the social distancing. It's very difficult to maintain because of the nature of the work and the work practice and how the production line is designed. It's not always feasible to ask workers to work back to back, side to side. And it's also difficult to put the screen or dividers between workers who work side by side because the design of the production line. And the areas have very high noise level, as I described. It caused problem in communications. And also people are not always wear the face covering. You can imagine that production workers wearing boots and overall aprons, visor, face covering, and the hearing protection is very difficult, and they still need to work as quite a medium physical activities, and they still need to interact with other workers and interact with people in the office area. So maintaining less than 2 meter social distancing is very difficult. And I also highlight issue with ventilations and also the unfavorable environmental conditions like low temperature, high humidity. Last but not least is the shift working, the night shift working. But I have to say that this workplace has controlled the outbreak quite quickly. And over time, I think they have a lot of control measure in place. And highlight their leadership is quite obvious. So I want to summarize that food sectors do have increased rate of COVID-19 outbreaks, but particularly related to the early stage of the pandemic. The relevant risk factors, particularly for the meat processing, because that's most of the evidence we gather from, including employee high density and also shared transport, and difficult to maintain social distancing because of the work practice, and I describe all the challenges and difficulties. And I think the environmental conditions may also play a part in the virus transmissions, and lack of the ventilations and shift working and linking with the community transmissions. I think that's the end of my presentation. Thank you.
Thanks so much indeed, Yiqun. Really, really interesting data. And it's prompted some questions from our audience, which I will try and cover very quickly. So first question is from Emma Agnew, and it was about the definition of an outbreak that was being used in the analysis that you talked about, and the definition that might be used by businesses. So what's the definition of an outbreak being used in your analysis and by business?
Yeah, we use the definition of outbreak defined by Public Health England for all our analysis, as well as for the COVID study. And we publish our research protocol online, so you can see all the link of the definition. And I have to say that definition is consistent applied through all our evidence review.
Thanks, Yiqun. The next question is from Noel Sykes and is about the spot inspection process. And I think actually Joe McMeekin has answered this to some extent in the chat. So if we can maybe pick that one up at the end, I think that will be good. There was a question from Karen Bavister about the equipment that was used to monitor CO2 levels. So could you, do you know what the equipment was, Yiqun, that was used there?
I don't know, and I did put into the notes in one of the presentation is that is using Honeywell, and I think I need a hygienist to answer that question.
Excellent. Well, if maybe whilst we're hearing the next couple of presentations, you could root around and see what we can find, that would be great, and then we can pick up on that in the chat. There have been a couple of other questions put into the chat, which we'll pick up at the end, but if I can encourage people to put them into the Q&A box, which is accessible from the bottom of your screen, that makes things a bit easier to follow, but I will pick up the question from Peter Noon later on when we have the discussion. Thank you very much indeed, Yiqun. Really, really interesting. And that brings us on to the next set of presentations. So I think it's going to be Rob first, and then Barry talking about some of the surface sampling that's been done during both the studies that Yiqun described and more broadly. So I think, Rob, it's over to you first. Thank you very much.
Thank you. I've got a few minutes really to talk about some of the practical aspects and some of the early work that we did in terms of environmental investigations on a number of sites, largely chilled food sites. And then Barry's going to go into the detail, what we actually discovered as a result of those investigations. Fairly early on, colleagues will remember, or you may remember, the quarantining of individuals in a largely, I mean, I'd say largely unremarkable place in the country, on the Wirral, in Arrow Park Hospital, but a place with which I'm, I've spent many an hour within the hospital, having come from the Wirral. So, but other than that, it did reach notoriety with individuals being quarantined, including those from the Diamond Princess. So that was at the start, I suppose, of the event. We didn't know a great deal about the transmission and in particular environmental aspects of transmission of this virus. So we then got involved in sort of workplace outbreaks, and I suppose just as a as an introduction to myself, working for the what was the Public Health England Food, Water, Environmental Microbiology Labs, our work with the food industry, with local authority environmental health, colleagues in other agencies, the HSE, the FSA, and other agencies, was largely and is largely looking for other targets. So our salmonellas, our listerias, and our legionellas in all sorts of matrices. But so this was fairly new for us in terms of the food and water microbiology labs and as outposted scientists. But I think what we're good at and what the technical teams are good at in these settings are pathways and understanding pathways, pathogen pathways, and making assumptions sometimes, which are often quite valid. So we had initially some queries from the incident management team about chilled food production and the risks associated with those sites. In particular, as many will know, that we have a large amount of recirculated air, chilled air in these sites. The refrigeration hasn't got the capacity and capability to take ambient air at 25 degrees, chill it down to all sorts, 3 to 5 to less than 12, the sort of chill temperatures, but to take 100% of the air, fresh air in, which was really what the guidance initially was about, sort of as much fresh air as possible. But we couldn't do that in these settings. So was the recirculation of chilled air, were we recirculating virus particles into production areas? So the incident management team asked a few pertinent questions, and without knowing the answers, we set off and tried to assist both the incident management team and the business in trying to identify any problem areas. So one of these key aspects initially was the air handling systems, but also we worked towards looking at key contact points. So there are a number of sites that we looked at, and this is thanks to colleagues in all sorts of sectors for their support and their patience with us on site. So we went to a number of different sectors, really to assist and to reduce that impact on the business and on the food chain. So we looked, as I say, we looked at the sort of handling units, and there were we took some swabs, some environmental swabs, and I'll show you the swabs in a minute. But these sort of filters, which will be familiar to many of you, are standard filters, so not high efficiency particle filters that we'd expect in some settings. For example, our hospital theatres, where we have these the sort of, we want a higher level of filtration. So we looked at these. We looked post-filtration, pre-filtration and post-filtration to see if we had significant virus particles there. In some settings, in limited settings, I mean, we didn't go to a huge number of places, we found that these filters were absent from the filter housing that they should be in. That's largely as you get condensation that occurs, the housing deteriorates, rusts, corrodes, and the filter housing, the new filter housing, was just placed on top. So the clearly the air pathway, the air is going to find that pathway of least resistance. So on occasions there was limited filtration going on; it was just being recirculated. So that was a maintenance issue that we picked up. But we looked in the these what I call SOCs, seen in all sorts of sectors now, to see if we could find the target that we were looking for, these gene markers for the SARS-CoV-2 virus, to see if it was coming back into our work workplace settings. So we were just recirculating. So looked at a number of these areas. We also looked at some contact points, and on the right-hand side, you know, we've got a door in a setting. It's a push door. It's required to be pushed all the time, and is a significant contact point. So in that particular setting, it assisted the business in going for rapid roller doors, which were appropriate. So pushed towards that sort of direction to reduce the contacts, and that review of some of those contact points was quite interesting. The swabs we use these sponge swabs. They're pre-moistened swabs. We use them for all sorts of targets, as I say, sort of Listeria, Salmonella, and for our indicator organisms often. We knew they'd worked because colleagues in biosafety had used them earlier on in the outbreak or the epidemic in other clinical settings, and we knew we could recover these targets, these virus targets from there. So it was a great swab to use. But we also looked at other contact points, and they're these largely unflattering photos of me, but we were looking in some of the non-production areas, so staff welfare areas, which were well segregated, but it's this review of the contact points was really important. That often the chairs and the table tops and sides, the tops are cleaned and were being cleaned thoroughly, but it was the underside, the side that you hold on to draw the chair into the table. They're the bits that we needed to focus on, and they're the bits that reminding that business looking at these pathways was quite important. On the right-hand side, the sort of biometrics again, so the signing in, the clocking in cards, again, an area which we don't take a great deal of heed to in terms of hygiene, because to get into production, we would then go through hygiene measures, hand washing and the like. But still an area of attention that we could focus on. So these sort of environmental investigations were to facilitate, I suppose, the incident management team, the incident control team in advising the steps that could be taken, but also helping the business in keeping operational and trying to identify key issues. And I'll pass you over to Barry.
Okay, thanks, Rob. So before we jump into the data that was generated from Rob's public health response activities, there is a second mechanism from which we collected workplace surface sample data. And this is through our research activities, primarily through the PROTECT study. Now, fortunately, Andrew has explained PROTECT to you at the start of this webinar, so I don't have to go into a great deal of detail on this slide. Safe to say that this work is within Theme 1, the outbreak investigations, and the main program within Theme 1 is COVID Out. So COVID Out is a program that's aimed to get some real world data from these workplace outbreaks. The first step is to identify the workplaces that might be experiencing an outbreak of COVID-19, and this is done through databases within UKHSA and the HSE. At this point, we will try and recruit these sites to this research program, which is by far and away the most difficult part of COVID Out. UKHSA and HSE have regulatory roles, and trying to convince these sites that this is purely a research study can be quite challenging. If we get consent from the sites, the next step is to arrange a site visit where a number of assessments will be carried out, including the surface sampling. And finally, the fourth step, the step that separates COVID Out from many of the other similar activities worldwide is the interaction with the workers, recruiting workers to the study, in particular with questionnaires, personal swabs and blood samples. And this is to get some high-level epidemiological data to provide further insight. And this is the element which makes this probably the most comprehensive analysis of workplace outbreaks in the world. And from this, we get an incredible amount of data. And for the rest of the talk, we're only going to be focusing on the surface sampling aspect of this, but COVID out is far bigger than you're going to see from the remaining slides. So the samples that are collected both from COVID out and from Rob's work, they both go through the same pipeline. So they're collected using these sponges or swabs, and then the material collected will be purified and have the nucleic acid extracted using commercial kits. And the purified RNA will then be analyzed using SARS coronavirus 2 specific assays via real-time PCR. And the benefit of this analysis is it provides not just qualitative results, the presence absence, but it also provides quantitative data, so we can see how much SARS coronavirus 2 material was there. So we're going to jump into the data now, and we'll begin with the sampling efforts that were led by Rob, the public health response activities. And from August 2020 to December 2021, 8 workplaces were sampled, including 4 from the food sector. And the take-home messages from this sampling is that we found 47 of these samples positive, which is just under a quarter. And a further 20 samples which were suspect positive. And this is where only one of the 2 replicates that are tested are defined as positive. And this usually indicates that the sample has a low level of RNA present, right about the limit detection for the assay. And just under 2/3 of the samples were completely negative for SARS coronavirus 2. Now, most of the samples that were positive were from high touch point areas. And overall, 5 of the 8 sites that were sampled had more than 20% of the overall samples testing positive. Before we go on, it's probably worth defining what we mean by positive. And this comes back to the qualitative versus quantitative. What does the value produced mean? And with real-time PCR, we get something called a CT value, a crossing threshold. And this is the number of cycles it took for the sample to be defined as positive. So the more material that's present, the less the number of cycles it takes for the machine to determine that the sample is positive. So the lower the number, the more RNA that's present. And we've known from extensive testing on not just these surface samples, but also clinical samples, that the CT value is quite indicative. And we know that if we get a CT value of below 32 cycles, we stand a good chance of getting whole genome sequence back from the sample. And if the CT value is below 30 cycles, that we stand a chance of getting the virus isolated from that sample. And obviously, the lower the number, the more likely it is that we're going to get this high level data back. But what is less certain is when we get these values which are above CT32, we're quite certain that these CT values of 35 upwards, these are very low level, right at the limit of detection. So this is a sign that contamination had occurred at this point at some time, but the level that was found does not present a realistic risk. But where we're less certain is in this 32 to 35 range, which is why it's coloured in orange. So if we have a look at Rob's sampling again, using this colour metric, what we see is that with the same 195 samples, 14 of them were in this amber coding, the CT32 to 35, and we had 2 samples which were in this red colouring, this lower than CT32. But it is important to note that both these samples only just crept into this bracket. Both were just above CT31, and indeed we didn't get any whole genome sequence back from these samples. So it's probably just about the exact positioning of this categories. So 92% of the samples that were taken by Rob were in this either negative or above CT35, but we did have 16 samples overall, which showed some significant level of contamination. With the COVID out sampling, We managed to recruit 13 sites from March 2021 up until last month. 11 of these allowed us on site to conduct surface sampling. There's a lot of data here. You don't need to focus on any of that. I'm going to go through the high level stuff, but it is important to point out that site that Yiqun discussed in the last talk, this is site 5 on this table. So 11 sites that were sampled, 4 of these were in the food sector. These are marked with an F, including site 5. We found 61 positive samples and a further 44 suspect samples from these 758 samples collected. So this is 8% positive and 5.8% suspect. 82% were completely negative. And again, this was mainly in high touch point sites, but we only saw 2 of our 11 sites where we had 20% or more positive samples. So when we have a look at the same data with the what was the level of contamination found, what we see is that we only found 15 out of the 758 samples, which is 2%, which was potentially significant levels of RNA, and none which had significant levels CT32 or lower. So we're going to have a look at some of this data in graphical form. I'd love to give some in-depth analysis here. We need to wait for the epi data before we can draw any hardline conclusions here. But just by glancing your eye over this, you can see this drop-off that occurred in late spring, early summer, where we were seeing far less numbers of positives recovered from each of these sites, although there were a couple of standouts in site 8 and site H, but these were still less than we were seeing in April 2021 and earlier. And also when we look at the areas that were sampled at these sites, I was actually quite surprised to see that there was quite a high number of positivity in all the types of areas that we sampled. And perhaps most surprisingly, the area which gave us the lowest number of samples is one of the areas that we most frequently target in outbreak investigations, which is the toilets. But one possible explanation why we were seeing a lower number of positives in this environment is this was an area which probably had a higher level of hygiene compared to many other areas that were sampled as a standard. And when we were adding these extra layers, some of the control measures were extremely elevated in these toilet areas, which is probably explaining why we're seeing lower numbers from that area. But again, we need to do a lot of epi data analysis to drill down into just how significant these findings were. So onto some overviews. What you've probably seen from those tables is there were some differences between the UK HSA-led and the COVID out sampling. More positives found with the UK HSA-led sampling and more potentially significant samples identified. And the reason for this is probably 2 important factors. And the first is that with the UK HSA-led sampling, they were able to get on site much quicker than we were with COVID out, because COVID out was a research activity. There was a lot more negotiation with the site, trying to convince them to participate and finding mutually convenient times. So we were probably getting on site 2 to 3 days after the UK HSA timeframe. But also vaccination status, the COVID out on-site sampling didn't start until March 2021. So we only had a couple of months until the majority of the workforce had received at least 1 dose of vaccine. The lowest CT value found to date was CT 31.07, which is a lot higher than I would have anticipated at the start of this outbreak. And that was from a food sector site. And we sent a number of the more intriguing positives that we found away for whole genome sequencing, and none to date have returned more than 50% sequence. So when it comes to the site-specific data, surface sampling can identify weaknesses in cleaning regimes and areas of increased risk. And the data generated has led to sites updated their procedures, which is one of the main benefits of COVID out so far before we've done this high-level analysis. And we found no samples to date inferring infectious viruses present at the time that we sampled these areas. More widely, there's an apparent decrease in positivity since summer 2021, and there's numerous explanations for this, including the increase in vaccination rates, and we need to do some more analysis before drawing firm conclusions. conclusions. But interestingly, there was a potential for location-specific risks to be overlooked in these areas, that there was broad-ranging COVID control policies which didn't change when certain areas within a workplace had higher risks of potential transmission. Just a quick slide on the legacy activities. This is one of the important factors of Protect, is this is trying to leave a legacy impact after this pandemic is done. There's a huge amount of research that's going on in the background on optimizing sampling methods, molecular methods, virology methods, to hopefully transition to other activities such as the sampling for endemic respiratory viruses or imported high consequence pathogens. And just some final thoughts to leave you with. The food sector has experienced impactful outbreaks, but they're not alone here. Many sectors have been similarly affected. Positive samples were typically in the non-production, the low care areas. In fact, we only found one positive sample in high care areas in the food sector sampling. And this is probably due to the elevated hygiene control measures that were already in place in these areas. Importantly, a site is not a single entity. It's a collection of different areas, and risk assessments and control measures need to reflect the differences between these areas. And it's likely that the impact of COVID is linked to interactions with staff and the infection control measures in those places. And this is where the difficulty lies, in balancing productivity with the optimal working environment. And finally, a big question which hopefully everyone is considering at the moment: what COVID control measures should be kept as we move into a post-pandemic world? There seems to be some thought that we should just be going back to the pre-pandemic world, but a number of the control measures that have been implemented recently are effective against a number of pathogens. So perhaps a better way to look at this is which low cost and easy to implement measures should we be keeping when we have a choice going forwards. And with that, I'll leave you with the acknowledgement slides, and I'm happy to answer any clarification questions that have come in.
Brilliant, thanks so much indeed, Barry and Rob. Really useful and interesting presentation. There is one question of clarification I'll take now, but I will pick up on the other questions at the end. And it's a question from an old friend of yours, actually, Anthony Wilson from the Food Standards Agency. And Anthony thinks he missed a step. He saw the relationship between the CT and the contamination level, but didn't understand how that then became an assessment of risk. Can you just explain that a little bit more? Yeah,
this is something where we don't have firm data to give you exact details. So the more viral materials present, the higher the likelihood is that there either is or was potential for transmission there. So what we are really looking for from a scientific perspective is these low CT values. This gives us a chance to potentially isolate virus, show that there was a contamination risk in this or a transmission risk in that environment. We haven't had any samples which have given us this definitive evidence. We know that this is the logical scientific method, that the more virus was present, the more RNA we will find. But until we can isolate virus, we cannot join these two steps together. And this is all down to timing of how quickly we can get onto site. We know that this will have happened at some point. What we're missing just from our data at the moment is this A to B link. But from a workplace perspective, it's reassuring that the virus doesn't seem to persist for very long in these environments. It's probably days to hours that the virus remains infectious. But yeah, that's the theory behind it. Thanks, Barry. We are limited for time, so what I'm going to do is pick up on the remaining questions at the end, and we'll now move on to Miranda Lowe. So Miranda, if you're ready to talk to us, it's over to you. Thank you very much.
Great, thanks a lot, Andrew. So I will just get going in the interest of time. The first thing I wanted to do is to thank the participants that we've had from the food and drink processing sector, either as participants in the study or those who have helped us kind of consult in terms of designing the study and giving us advice. So it's been a challenging time, so we really want to show our appreciation for their effort to help us in this research. And I just want to point out that the work that I'm presenting today is really not my own work only, but we have a research team from the London School of Hygiene and Tropical Medicine, IOM, as well as the University of Manchester. And so I'm really representing the work of this team. So just a little bit of background. So we also did a review of sector risks in the food and drink processing sector. And generally speaking, at the time when we did the review, there was quite limited literature in the UK sector. Most of the evidence seemed to come from the United States, and especially from the meat and poultry processing facilities and plants. So some of the factors that were highlighted in a review as being potentially important for increased transmission risk are socioeconomic factors such as ethnicity and low income, as well as lack of sick pay. Environmental factors also were pointed out, especially due to low ventilation and a lack of social distancing in food factories, and therefore potential higher risk from aerosol transmission. There's been kind of a lot of anecdotal discussion about the role of shared accommodation and transport to and from work, especially given that the workforce could potentially be made up of a fairly migratory group of people, but there isn't necessarily clear evidence for this. So with this in mind, we also remember the presentation that Yiqun just did a little while ago, and there did appear to be in England a fairly high outbreak rate in the food sector. So this points to kind of wanting to understand a bit more about what's happening in the UK in the food sector. So in theme 3, we undertook to do a kind of a sort of mixed methods study to better understand the food and drink processing sector specific risks. So in addition to the literature review I mentioned earlier, we also looked at national statistical data from the Office of National Statistics to ask whether risks are elevated relative to other occupations in the food and drink processing sector. And then we also worked with the sector and company representatives to understand better through survey and interviews what the impacts have been in their workplaces, as well as what they perceive as being what they perceive as being risk factors in their sector. So this is some of the work that we've done based on the ONS data. So this is actually a summary of different studies first. And the top line is the work that we have done, which I will go into a little bit more detail, but I just wanted to show here that these are all UK-based studies from different data sources. So we have the ONS, the UK Biobank, as well as a separate data source from the ONS and the VirusWatch study. And generally speaking, what we found is that the risks are not necessarily very high. And if I just give a quick primer on how to read this kind of graph to those who may not be familiar, if you look at the red line, that essentially at 1, that essentially says that if you are at that line, there is no risk relative to the comparison group. So we're comparing food and drink production sector to non-essential workers, or say professional and associate workers in the case of VirusWatch. Now the lines, so the dots will represent the best estimate of the risk, and the lines indicate that, as I mentioned, it's the best estimate of the risk. However, there is some uncertainty around that estimate. And so the line that shows you the extent to which that estimate might in real life be. So that's the estimate, the range where the true risk may be. So you can see here that in the ONS infection survey results, which is the first line that I will discuss a little bit more, the risks are slightly lower relative to non-essential workers, whereas in the others they're slightly higher, but all of them more or less, the lines will straddle 1, which show that it's not necessarily a significant risk in the sector. It's a statistically significant risk, I should say. So just to go back to this slide, quickly speaking, in this, compared to other workplaces, so we compared the ONS data that I mentioned earlier, there actually is, compared to say drivers or taxi drivers, bus drivers, they are much higher risk. And if you consider that their risks actually are clearly above 1, so there is a clear higher risk in other sectors, education as well. So comparatively to non-essential workers, these other sectors of essential workers. So if we're comparing across the groups of essential workers who have worked throughout COVID, food sector appears to have a lower risk compared to things like public transport, as well as education, etc. So the question then is, how did the sector experience COVID in practice, and how have they mitigated the risks? So the next part of the study sought to understand this. I'm sorry, there's a bit of a mix-up on my slides. So this is the ONS, more details on the ONS infection survey that I had mentioned earlier. So you can see the details here. So the other sectors I had mentioned before, the taxi cabs, coach and bus drivers, etc., they had consistently higher risk. And please remember that this is kind of averaged out over a fairly long period of time, from April of 2020 at the beginning of the pandemic to October 2021. So at different stages of the pandemic, these risks may have changed, with, for most sectors at the beginning, there being relatively higher risks. But you can see that generally speaking, as an average, food production is lower as an essential work sector than the others. However, I'd like to point out that this is based on the infection survey, and the infection survey is based on household addresses. So people come to visit people at their household address. So as I was mentioning before, since the sector does tend to be quite dependent on migrant workers, it is possible that it's an underestimate of the actual risk, just because these people may not be fully captured within the infection survey compared to those in other professions. Okay, so apologies for that slight mix-up in the slides, but to really kind of explore these risks in a bit more detail, so that was looking at kind of all professions in the UK on our UK-wide data set. We wanted to kind of work a little bit more with the representatives of the sector. So we have designed the COVID at Work study, which consisted of 4 stages: a baseline survey, which is done online, a follow-up telephone interview with companies in the food and drink manufacturing sector. We also did a sector level study that was a qualitative study based more on interviews with people who are either experts related to the food sector, such as scientists and academics, people who worked in policy, regulators, trade associations, as well as unions. We have not yet done a stage 4. We're still in the stage, in the practice of developing this part of the study, which is looking a little bit more at interviews with employers and employees at companies, etc. I'll say a little bit more about that at the end. So if I begin with the baseline survey results, this is just a summary of what we did. It was, as I mentioned, an online survey, and we were really looking at kind of recruiting companies into the study at this point. So we engaged with various food and drink manufacturing federations and associations to try and encourage dissemination of the survey to the members. We ended up with about 33 companies completing the survey, and this was represented 66 sites that were located throughout the UK. And you can see the distribution of the subsectors that were represented within the participants. This covered the period, so the actual survey was rolled out in early 2021, but we asked them to look back till March 2020. So the data that was reported will cover that period of time. So we looked at the results from this baseline survey. The graph on the right is similar to what I had showed earlier. So one being there being no risk reference in comparison to the reference group, which in this case was the beverage sector that had answered our survey. So you can see the distribution. It would appear, compared to the beverage survey, that grain milling storage had a higher risk than, say, manufacturers of prepared meals. But again, this is relative to the beverage sector. The overall message here is that we did find that, as you might expect, those companies that were able to have more people working remotely, and also where their workforce perhaps were a bit better off socioeconomically, they tended to have lower risks, and those that had a larger number of workers that had to be on site at any given time would tend to have greater risks. We also found that the risks tended to follow the background rate of risks in the community, meaning that overall it appeared that in the companies, the rate of risk would follow what the community that the company was in, the risk that they would have over time. So just to go to the follow-up survey results then, again, this was then done 6 to 9 months after the baseline survey. And here you would see the a scatter plot with on one axis, the x-axis being the stage 2 monthly rate per workers, rate of infection, and then the stage 1 is on the y-axis, the vertical axis. And it would appear that there were higher COVID-19 rates compared to the baseline in the second stage. We aren't 100% sure if this is due to increased testing, because as you know, as the pandemic went on, there was testing either around outbreaks, but also some companies did a little bit more regular testing as a prevention measure. In terms of the mitigation measures we asked about at this stage, so we asked a fairly large list of mitigation measures, whether they had done them, and it just appeared that most of the sites had actually implemented most of the surveyed mitigation measures. So you can just see in this graph here kind of how many measures were done per site, and it was not a very large variation in the number of measures. Most of them had a similar number out of the ones that we had asked about. So to complement this, we had done the sector level study, and these were interviews with, as I mentioned before, various groups. And there were 21 semi-structured qualitative interviews done with 32 individuals. So some of them were not individual interviews, but group interviews. And you can see that here the distribution of the different types of groups that we, people in different groups that we interviewed, and the interview transcripts were analyzed to see if there were any emergent themes amongst them. So what we learned from these interviews are the risk factors that our participants cited were the requirement for site-based work in the food and drink manufacturing sector. Obviously, this is a sector where it's difficult to work from home. You can't really produce sandwiches only from your home. So this is something where people had to go to work. And because there's a large reliance on manual labor, obviously this will vary within the industry, with some types of subsectors being able to automate a bit more than others. They had to have quite a large number of people on site. The rates, the speed of production was quite large. And therefore it's difficult to have, you would just have to be near people in the workplace. Social distancing wasn't always very easy. They did mention that cold ambient temperatures and humidity was potentially an issue because chilled factories tended to have large numbers of workers, and the temperature controls could make it difficult to ensure adequate ventilation. Infrastructure buildings, some buildings were older and therefore perhaps not as conducive to implementing some types of measures such as ventilation and social distancing. And also there was mention of spread in non-operational environments and shared facilities such as break rooms, as well as transmission from outside of the work environment. Generally, the industry felt that they didn't necessarily have an elevated risk compared to other industries that would have been working at the same time, so other essential industries. And their familiarity with having to use PPE to risk assessments, et cetera, and they have to follow hygiene standards means that they felt they were at an advantage with respect to being able to respond to the pandemic. The mitigations most commonly cited, again, these were also, we found in our survey, cleaning and hygiene practices, social distancing, measures to limit contact, as well as ventilation, testing and monitoring, as I mentioned before, as well as increased use of PPE. Generally, they felt that the level of compliance were fairly good, but they did feel that sometimes social distancing, both within and outside of the operational environments, may not be as good as it could be. So just a quick summary. I mean, there were a lot of challenges at this time. There were obviously, you know, this was a time when everybody was experiencing the challenges of COVID as well as Brexit. And so there were a lot of labor shortages, supply chain issues, and again, work-related challenges related to having a fairly low-paid staff and people who may not speak English as a first language. And so communication was sometimes something that they had that could have been a challenge in responding to the pandemic. I just want to conclude then with a summary of the things that we found. So overall, when we compare the food processing sector as an essential sector, the risks compared to other essential sectors were not necessarily higher. It is possibly more prone to outbreaks because of the nature of the work. Most of the facilities tended to have implemented a similar set of mitigations, but they did find that the shifting rules across the UK, as well as time went on, was sometimes challenging to respond to. As I've mentioned again, socioeconomic factors could be pose challenges for the response. And there is still some uncertainty about the contribution of workplace versus work-related factors, and work-related factors meaning things that might be associated with work but aren't at work, such as living in shared housing because you've got a migratory workforce or shared transport to work. And again, with Brexit as well as COVID, this was quite a challenging time for the industry. So we're aware we have a fairly small representation of the food sector in the COVID at Work study. And so, but given the challenges they face, they may have just had less time to engage in the research. So just to say something briefly before I end about the stage 4 that we're in the process of setting up. So as I mentioned before, we've looked at kind of industry groups, unions, academics, and government viewpoints on the food and drink processing sector. We'd like to get an idea in the next stage of how the pandemic is evolving and how this is experienced by the sector, especially from the workers' perspective. So if anybody on this webinar works in the sector, please do get in touch, and we'd be happy to speak with you a bit more about, you may participate in the development of the next stage or be a participant in the next stage as well. So there's an email address there in case you're interested, and the website is also there on the slide. And I will end there. Thank you very much.
Brilliant. Thank you very much indeed, Miranda. Again, just adding to our sort of knowledge base in this challenging area. And I think, you know, one of the things that's come out for me from all the work we've done in this space is the complexity of the system that we're operating in. And you know, what I think Protect has been able to do is unpick some of that complexity and see how that basket of risk factors, as I think Yiqun described them, can come together in different geographies and different locations to produce the outbreaks that we've heard about today. So thank you very much indeed for the 3 presentations. We've got quite a few questions, and I'll apologize to Paul right at the start because his has been waiting there for some time. And Paul makes a really interesting point about humidity and the fact that over the lifetime of the pandemic, this has become a bit conflicting. So Paul is keen to know if the consensus now is that high humidity is a greater risk than low humidity, and is it material? I don't know if Barry, you wanted to address that one first.
Sure. So humidity isn't my specialty, although it obviously plays a big part in a lot of the work that we do here. What I am confident in saying here is that there seems to be like a Goldilocks effect. It's not high humidity or low humidity, both are not particularly great for viral survival. It's the middle part which sort of determines how well the virus survives. I'd have to go and speak to some of my colleagues to get the exact data, but if it is too high or too low, it will have a negative impact on viral survival. We have done quite a lot of work on this. It does need to be in, I think it's about 40 to 80% range for the virus to survive any significant amount of time. But if it dries out too quickly, then there's no infectivity from the virus.
Thanks, Barry. Any other comments that people want to make on humidity? Whilst you're thinking, I'll just make the point that Barry, Yiqun, myself were at Harwell a couple of weeks ago, and they're doing some fascinating work there, using laser-based systems to levitate droplets, individual droplets, and visualize what's going on at the level of the virus itself. And I think they are looking specifically at some of the impacts of humidity on the behavior of those droplets in different circumstances. So I think we will be able to get a bit more information once those incredible levitating droplet experiments are completed. Any other views from Rob, Yiqun, or Miranda? Okay, let's move on then. And a really good point from Chris in that, recognizing that these studies are really hard to do. And I think both Miranda, Barry, Rob, and Yiqun have all described the difficulty of doing this work. So what could we do differently in a future pandemic to enable us to be on the front foot rather than the back foot? And I'll go to Yiqun first on that one.
That's a really hard question to answer. Yeah, I think getting the right evidence is quite important. I look at the literature review about how meat processing plan and react to the outbreak, and I feel like now the sector are more confident to control the outbreak. For example, at the beginning, temporary closure is one method to control the outbreak, but now it's more looking at the vaccination, testing, contact tracing. So I think for me, maybe getting the right evidence and pulling different expertise together and would be helpful. Thanks.
And I guess as well that one of the challenges here is that actually going to sites that are going through a pretty traumatic time and getting them to do something else at the same time as that, intervention from UKHSA or HSE is a bit of a challenge in its own right as well. But interested to hear Rob and Barry's views in particular, who've also been involved in going in on the ground.
But I think at the time, frozen now, we almost needed a guidebook for sectors, different sectors, to assist them a sort of pathway into what they should be doing, what actions should be taken, and something that's pulled together on the evidence base that we develop out of what we've done, so that when we get something that may recur, we've got something to hand that's that we can use really. And we can get up to speed quickly, whether that means closing for a short period or putting other interventions in place. Because we had a lot of questions that came through asking what should be done from all sorts of sectors, and having some guidance is really useful.
Thanks, Rob. I'm going to go to the next question, if I may, from Irma, which is about the rationale for splitting the MHR by male/female. Miranda, I don't know if that's one for you.
Sorry, the MHR being?
I'm assuming that it's one of the outcome measures from your slide, but I may be wrong. I don't know if Irma wants to just expand the MHR acronym, mortality hazard ratio.
Oh, right. Is she referring, I assume, to the ONS study?
Yeah.
So the ONS study, sorry, I'm going to just look at the question again a bit more detail. I'm trying to remember. So there wasn't mortality for so we looked at the infection survey. So that wouldn't be mortality. In terms of male versus female, it was adjusted for age and sex, I believe, just because those are things that we think might be potentially predictive of risk. And so we want to kind of control for that in the analysis. And I'm just assuming that's what she's asking about.
We'll wait for Irma's response in the questions. But let's go to Joanne's question. So this is about temperature control. So can we explain a bit more why temperature controls mean that you can't achieve adequate ventilation? I don't know if that's one for Barry or Rob.
I don't know. I was going to just give I suppose, some anecdotal experience. These chilled production sites are, as we all know, are huge sites, and we're dealing with production areas of, as I said, sometimes 0 degrees up to sort of 5-8 degrees, depending on where we are in high care or low risk areas. The plant that's typically installed to provide this chilled air relies on taking chilled air from production, filtering and sending a large proportion back in, and that's sort of balanced with a certain amount of fresh air. If we say in the summer try and take 100% fresh air at dare I say it, 25 degrees for summer temperatures through the plant hasn't got the capacity to actually chill the air down to the temperatures we require in the production area. So largely we take in some fresh outdoor air at sort of 25 degrees, and we recirculate the chilled air, and that gives us that sort of chilled environment that we're after. Does that make sense, Barry?
I don't think I have any more to add to that. Yeah, it's a matter of efficiency and cost at the end of the day, isn't it? If you had infinite money, you could devise a much better system, but you have to be practical with all these things, and there is a lot of recirculation that is ongoing in these places.
Thanks, Rob. Thanks, Barry. We've got a question from Paul about the fact that SARS-CoV-2 is clearly airborne, although we know that other routes may also play a role, and therefore adequate ventilation is crucial, and the sector predominantly has low ventilation or high levels of recirculated air. Are we going to be more prescriptive in the advice that we produce as a consequence of the work that we're doing to enable industry to perhaps work on different standards moving forward? And whether that might include UVC decontamination systems as well. Rob, Barry, Yiqun?
Okay, I'll kick off here. Yeah, there's nothing unreasonable in that. It comes back to the same point. We've got a lot of research ahead of us here, especially with things like UV decontamination. I think everyone will agree that we've got big strides ahead of us in terms of ventilation that can be provided. It comes down to cost again. But yeah, it's definitely something we need to improve on going forwards.
Thanks, Barry. Question from Hilda about the removal of removement of mitigations when the pandemic is still active, and health and safety law requires assessment and preventions under control hierarchy. So I'll go to Miranda for that one, because Miranda, you've obviously spoken to a large number of people in the sector, and interested in what your views might be on their views about the sort of change from a very prescriptive approach to one that is not so demanding in terms of the requirements that are needed.
Yeah, so I think, during the study, we still sort of had the pandemic is sort of ongoing. So one of the things we do want to look at now is, as restrictions are sort of pretty much gone, at least in certain parts of the UK, and will eventually be soon. We would actually like to return to some of the people we've interviewed, just to see what have they done or what has the perception been since then. In terms of when we were doing the study, there were some obviously restrictions where it was difficult for them to implement. But again, as I mentioned, they didn't seem to have they didn't necessarily seem to have an issue with implementing a lot of restrictions, as evidenced by them agreeing to a lot of the number of suggested ones that we had provided to them. So it's quite an extensive list, and they all had said they had done anything from the social distancing to trying to improve the ventilation where they could, putting up barriers. I think some of them did do CO2 monitoring. So I think what we are really curious to see is, do they keep some of these measures in place afterwards, just because they perhaps are not really interfering a lot with the work, and which ones do they actually end up removing? And back to the question that was asked earlier about, what do we do for the next pandemic? I think it's quite informative to understand sort of not the efficacy, but the ease of certain types of measures, and perhaps that could form basis of some guidance for the future, so that they can have something at the ready to do.
Thanks, Miranda. Really, really important point to end on. Hopefully what we've been able to do today is give you a bit of an insight into the work that we've been doing through the PROTECT study. Hopefully that has enabled you to see how we've used real world situations to gather really important data. Going forward, obviously what we want to do is to package that appropriately so we can make it available to people to use in their particular circumstances. I think as well, the complexity of the situation means that we can probably think about how, when you take COVID out of the equation, the information that we've collected will enable us perhaps to think slightly differently about health consequences and work more generally. And that's something that we're certainly starting to think about in my area going forward. So thank you very much for attending. Hopefully it's been useful. If we haven't had any opportunity to answer all of the questions, I know there are a couple we haven't probably addressed, we will get back to you directly. And we will make sure that we answer those. Thank you very much for your attendance. Thank you very much indeed to the speakers, and have a great rest of the day. Thank you very much indeed.